Hello and welcome to Nikolai's Genetics lessons, and today I have prepared two questions for you. Those questions are very easy. I still recommend you to watch this video up to the end, even if you know the answers, because I believe I would be able to provide you new information that you didn't know before. So the first question is: DNA is made up of repeating subunits called, and here four answers to choose from. Answer A, RNA, B, nucleotides, C, genes and bases. So let me explain every term here. So let's start from the end of this list. Bases, and bases, this is double-stranded DNA as you see, and bases would be this part of the DNA. So thymine, for example, would base pair with adenine, and guanine with cytosine. So this part we call bases. And what we call nucleosides, we call bases plus sugar. In the case of the DNA, sugar would be deoxyribose. In the case of the RNA, this is going to be ribose. So sugar and base we call nucleosides. It is not on this list, but I just want to tell you the difference so you would know. And the other answer would be genes, and genes would be large stretches of the DNA, and this is not going to be smallest subunit of the DNA. So we can cross out answer D, answer C, and we're left with two answers, RNA and nucleotides. And RNA would be transcript of the gene, so we can cross this answer also. So the smallest subunit would be nucleotides, and nucleotides would be a base, a sugar, and phosphate group. Once again, in the case of the DNA, this is going to be deoxyribose. In the case of the RNA, this is going to be ribose. So three elements that would make one subunit that we call nucleotide. So our choice would be answer B. And now let me go to the second question or statement. In the United States, it's currently legal to patent naturally occurring genes, whether it's true or falsely. So until 2013, to be precise, until June 13, 2013, it was true. In United States, a company could patent genes that were naturally occurring, whether in humans or animals or plants or bacteria. But in 2013, it was ruled by Supreme Court of the United States in a case against a company that's called Myriad Genetics. That company held patents for two genes, human genes, or mutated alleles that we call BRCA1 and BRCA2, that were used for diagnostic purposes in order to find predisposition of the person who has these mutated alleles for the breast cancers. And this company charged unrealistic prices for other companies to use these alleles in order to diagnose breast cancer. And as soon as Supreme Court made its decision that no naturally occurring genes can be patented, prices dropped down from $4,000 to less than $1,000. But there is not the whole story. Imagine that this is messenger RNA, and messenger RNA code for some protein, and when it's made, first it consists of the fragments that we call exons and introns, and introns would be cut away, and the final version would be made that would consist only exons. So this fragments would be cut away. And in biotechnology, this messenger RNA, mature messenger RNA, can be used in order to make a complementary DNA. So, using reverse transcriptase, this enzyme would make another DNA strand, then this messenger RNA would be degraded, and second strand of the DNA would be made. Now, as you see, this double-stranded DNA would code for the same gene, but wouldn't include introns. And this fragment that has a whole gene without introns can be inserted in bacteria DNA. So imagine that this is circular bacteria DNA. It would be cut and new fragment would be inserted here. Why we do not insert in bacteria DNA whole human gene that also includes introns? Because bacteria cannot cut away introns, because bacteria normally doesn't have introns or just very few of them. So this is what have to be done. This happens naturally in our organism, but Supreme Court leave this opportunity for biotech companies to use these genes as not naturally occurring, that this is something that they invented, but this is not so. This sequence happens only in messenger RNA, but using this technique as I described, scientists can insert this double-stranded DNA that code for this gene for certain protein. For example, it can be insulin, and bacteria would be able to produce this protein that can be later refined and used in order to treat human genetic disorders, like lack of production of this enzyme. And if you would take a look at this picture, you would see that the same amino acids can be coded by different sequence. So there is also another opportunity for biotech companies to change sequence of the DNA without changing the sequence of the protein. For example, if in certain place in human genome or human gene we have uracil, 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 scientists can change it to the uracil, uracil, and cytosine, and still the same amino acid would be produced. So this is going to be another way how biotech companies can overcome restrictions of this law. And also I want to tell you that, for example, in bacteria, the same amino acid in most cases can be, for example, coded by this sequence, and in human genome, say in 70%, would be used this trinucleotide, and bacteria would use in most cases this trinucleotide. So in many cases scientists need to change nucleotide preference in order to express this gene in bacteria genome. Otherwise, if the sequence wouldn't be adapted, whether it wouldn't be expressed at all, or production of the protein would be very small. So as you see, the first steps are made in right direction, but still biotech companies has a lot of different ways how to cheat and overcome restrictions of this law. But I hope that in future we would see that there would be more and more restrictions how biotech companies can use naturally occurring sequences that code for the proteins. So as you see, the correct answer would be answer B. But as you see, this is not as straightforward answer as we would prefer it to see. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Please share this video with your classmates, and see you in the next video. Goodbye.